All right. Hello, everyone. This is a topic for 5.4, non-Mendelian genetics. And so where we left off in topic 5.3, we were basically talking about Mendelian genetics, which is just like kind of more conventional. How do genes found on chromosomes in the form of alleles move from parents to offspring? And so these are things that don't fit that mold, that kind of go more exceptions to the rule. And so they don't always follow this Mendel's law. A lot of them can be more complex. And so genes that are adjacent and close to one another is going to be a big theme in this topic. And then so they're found on the same chromosome that are inherited together, and these are known as linked genes. And so these are genes that are generally inherited together. And so we'll talk a little bit more about that. But basically, a common example you can think about is red hair and freckles. People with red hair typically also have freckles. And what we found is that those genes are found really close together on the same chromosome. So most of the time, they are being inherited at the same time. And then we also have traits that are determined by genes located on our sex chromosomes. These are chromosome pairs number 23. These are known as sex-linked traits. And so linked genes and sex traits do not follow the ratios associated with Mendel's laws and can be identified by quantitative analysis. So this is just a fancy way of saying we could do like chi-square. We could do probabilities. We're basically looking for expected ratios or expected outcomes and comparing it to the actual outcomes and seeing if they match up or not. So some things that follow the non-Mendelian genetics that go kind of do their own thing or follow their own modes of inheritance. First is incomplete dominance. So this is when phenotypes for two alleles are blended together. Now this was the prevailing thought for how traits are inherited by people before Gregor Mendel, is that there is a blending of traits. You know, you get a short tail and a long tail, and you get some kind of tail in between. Now, I found that's not true for most genes. However, there are still some genes that can show this incomplete dominance. For example, the flower color over here, we have a red flower, we have a white flower. When we cross them, we do a cross-pollination, we get a pink flower because those colors have blended together. Next one is called codominance. And so this is when it's different from incomplete dominance. This is when both traits are being shown equally. And so we can see this with blood types. And so if I draw some blood cells real quick, let's say we have blood cell A and blood cell B. Blood cell A will be, maybe they have spikes on them, which makes them that A type. B has some squares on them. And then if we look over here and look at AB, we can see that codominance is happening because they have both spiky and square receptors on their membranes. And so codominance is when both traits are expressed equally. We can also see this in a few animals, you know, like when we have stripes in animals or cows with their kind of like black and white pattern. That would be an example of codominance. And then the next one is multiple alleles. And so this is when there are more than two alleles that can be expressed for a trait. So typically we talk about there's like big A and little a. In multiple alleles, that's going to extend past those two. And so blood types, you can be type A, you can be B, we just mentioned you could be AB. And then the fourth blood type you can be is type O. And that's when you have nothing on the outside. There's no receptors. This is just empty over here. Okay, and so this is going to help determine what type of blood type people have is based off of what receptors are found on the outside of their blood types. We also have a thing that goes into blood types, our H factor. It's a type of protein on the outside of your red blood cells. So you can have, you can be like A positive, B positive, AB positive. That's referring, the positive is referring to having those proteins on your red blood cells. And then B negative means you just don't have those proteins as well. And so AB positive, you'd have, not only would you have the spikes and the squares, but you would also have all of these other proteins on the outside. All right, so I want to spend some time to talk about linked genes. So these are genes, like I said, that are adjacent and close to one another on the same chromosome. Okay, the big idea here is that they're found on the same chromosome, and they may appear to be genetically linked. And so these are typically inherited together. And so linked genes are less likely to be separated during crossing over in meiosis. Because they're so close together, there's less ability for them to kind of be separated. Right? If we have a gene over here and down below here, those two are going to be separated pretty easily. However, if you have a gene here, you have a gene here, these are going to be what we call linked, because it is going to be very difficult for crossing over to separate those two genes when homologous chromosomes overlap with each other during meiosis. And so we can use a unit to kind of detail how far away genes are from one another, and so it's called map distance, and tells you how close together a pair of linked genes are, determined by how frequently a pair of genes participates in a single crossover event. And so linked genes have a recombination frequency of less than 50%. So if we have like a chromosome, 50% is kind of our threshold for when we decide if it's linked or not. Because at a certain point, if they're too far away from each other, like genes A and B are here, then they are not considered linked. They are just inherited separately and have no relationship with each other. And so the lower the number, if they have a recombination frequency of 5%, they're going to be close together on that chromosome. And so 5% we can also translate to 5 map units apart. Okay, or what you'll see is like MPU. And so if they have a recombination frequency of 30%, they are further apart on that chromosome. Okay, and so we can use this to determine the distance between genes and on a chromosome. And so we have like an example problem down here. We have genes. I wrote out a few different types of genes, A, B, C, and D. And then I wrote down some recombination frequencies, meaning how often do they cross over with one another in a recombination event. And so what we want to do is, I just drew a chromosome off to the right. We want to be able to figure out the order that the genes are found in. And to do that, we just have to kind of like map it out, right? These map units. And so I can just kind of arbitrarily say for right now that maybe gene A is right here. And then I know B is 22% or recombination frequency. So we can also just say that's 22 map units. And so I can kind of do this. Note the chromosome is not drawn to scale. That is okay, since 22% is our highest number. So A and B are 22% apart. I can also see that A and C are 9% apart. And then let's see, what's else? A and D are 17 map units apart. And you might be wondering, like, how do we know if D goes on this side or if D goes on this side? Well, right now we don't. We don't know which side that it goes on. So all we can do is kind of like put our numbers in. It's trial and error. But basically we have to satisfy all 5 of these numbers. And the only way to do that is by putting them in the correct order. And so what we'll see is if I put D here and kind of say this is, where'd it go? 17% apart. Then we can look at the other ones. So B and D should be 5 apart, 5%. And that would make sense with us, right? Because A and B are 22% apart, A and D are 17% apart, so D and B should be 5%. So that's good. You can kind of treat this as like a checklist. And then the last one, C and D should be 8% apart. If we look at that, C and D are 9% apart, or A and C are 9% apart. C and D are 17% apart, or A and D. And so the difference is 8%, which should be our answer. Right? So gene mapping is a way to kind of visualize our genes and where they are on the same chromosome. Again, highlighting the sameness, the fact that they're on the same chromosome. And we can kind of predict how closely together they are linked by how close together they are. Okay. And so let's go back to sex-linked traits, or found on the sex chromosomes. And so sex-linked traits are located on these sex chromosomes. Again, chromosome 23 for humans. They determine biological sex in animals. Sex chromosomes are non-homologous. Okay, so every other chromosome in our body, 1 through 22, are homologous pairs. Chromosomes 23 are not. Our X chromosome kind of looks like a standard, typical chromosome. And then the Y chromosome, you can see, is very non-homologous. It's much smaller, not as many genes on them. And so it's not every animal is X and Y, or XX or XY. There's other sex chromosomes that we use for different animals. So just be on the lookout for that if you see them in problems. And then sex-linked traits deviate from Mendel's mode of inheritance. So they don't necessarily follow that dominant recessive autosomal mode of inheritance that we've been seeing. And so sex-linked traits or pedigrees have a particular pattern in humans. Females are XX, males are XY. The Y chromosome carries very little genetic information. Therefore, most sex alleles are only carried on the X chromosome. We basically want to just assume that when it's a sex-linked trait, it's going to be found on the X chromosome. The only exception is when we find these like pedigrees that have only the males and all of the males carrying a certain trait. Then that might be cause for making a claim that the trait is found on the Y chromosome. But we might see this in problems too, where sometimes they like throwing some curveballs. If those traits are found on the Y chromosome, that's actually called a holandric inheritance. So holandric is just on the Y chromosome. And so over here we have some possible genotypes for our conventional sex-linked traits. And so notice we just always include the sex chromosomes. XX, this is a female, these are all females. XY are our males. And so notice for our males, we only have one allele on our pair of sex chromosomes, and they're only found on the X chromosome. The Y chromosome is not carrying anything in here because they are non-homologous. And then going back to holandric inheritance, some things that are found on the Y chromosome. So this is where biological sex is determined for males, for testes formation. You could also get webbed toes from the Y chromosome. Hypertrichosis of the ears, excessive hair growth, or ichthyosis hystrix gravior, which is a strange thing. I'm not going to Google it or show you, but if you want to look into it, it's called porcupine man. It's a very interesting and rare genetic disorder that you can inherit. And so our alleles for sex-linked traits are shown as a superscript of the X chromosomes, X big A, X little a. Females can be considered carriers, and that's just referring to when females are heterozygous for a recessive sex-linked allele. And so what we'll see is that color blindness, for example, is a recessive sex-linked allele or trait. And so females can carry it without expressing that color blindness themselves. Males are more likely to have phenotypes associated with recessive sex-linked alleles. And so that's kind of a good thing to consider when we look at like pedigrees, is that if males have it more often or in higher frequency than females, that might be a cue to be a sex-linked recessive trait. All right, so let's take a look at some examples. The pattern of affected offspring with unaffected parents. And so we have some individuals that are affected with something. Males are more likely to be affected. We can see all of the people that are shaded in are males in this pedigree. Sometimes you'll see pedigrees that have females shown as half-shaded circles. I wouldn't count on it, though. Typically, it's kind of depends on the problem. And so if we kind of go through this, we can kind of see, okay, if this is a sex-linked recessive trait, the male would be XY, and that would be big A, not little a. Female over here, XX. We don't know if there are homozygous dominant or heterozygous, but if you look, the fact that males are inheriting it would tell us that they are heterozygous and that the female would be considered a carrier. And so males, they always get their Y chromosomes from the dad. And so the male or the dad is determining that Y chromosome inheritance, and then their X chromosomes are always inherited from their mom. And so you can see that males here have a 50% shot of inheriting the color blindness allele. And so in this case, these two males inherit the recessive allele and are colorblind. This male on the far right inherits the dominant allele and has normal vision. And so we can kind of use pedigrees and our idea of sex chromosomes to determine how often or where and how they're inherited from their parents. All right, and then keeping up our trend with non-Mendelian genetics, our next one is called polygenic traits. These are traits that are the product of multiple genes and or physiological processes happening in combinations. That's a fancy way of saying that there are more than one genes that go into a phenotypic expression, right? You can think about like hair color or skin color, for example. It's not just one specific gene found on one specific chromosome to get your skin color or your hair color. There are many genes on different chromosomes that can determine it. So it gets very complex pretty quickly. That's why oftentimes we are not able to completely predict phenotypic outcomes of offspring just by looking at their parents, right? It's not that easy most of the time. All right, and so sometimes polygenic traits, the dominant allele codes for dark pigments, a recessive allele does not. And then so the figure below shows possible genotypes and phenotypes if hair color was coded for by genes A, B, and C. And so you can see you can get a lot of different types of combinations here by all of these different genes. All right, we have 3 genes here. Each gene has 2 different alleles, and so you can kind of start doing the probability of how kind of complicated or complex that can get pretty quickly. All right, and then the kind of last concept for this topic, some traits result from non-nuclear inheritance. Non-nuclear inheritance, what does that mean? So these are genes on DNA not found in the nucleus. Well, for humans or animals, what does that mean? Where else is DNA found? That's referring to the mitochondria. And so chloroplasts and mitochondria contain their own non-nuclear genome, right? Because they used to have their own self-surviving organisms. They used to be prokaryotes on their own that got engulfed by eukaryotic cells early on in life's history. So they have their own DNA and they divide separately from the cell as a whole. And so chloroplasts and mitochondria are randomly assorted to gametes and daughter cells during cell division. And so egg cells, what we'll see, are much larger than sperm cells. And so the egg cell is what's going to hold all of the organelles and all of the material that the eventual zygote will need to start dividing and becoming an embryo. And so mitochondria are transmitted to the egg and not sperm in animal cells. So what this means is that mom is passing on mitochondrial DNA. So we say that they are maternally inherited. Mitochondria and chloroplasts are transmitted in the ovule and not in the pollen in plants. Pollen is the sex chromosomes or the male sex chromosomes for plants. Females is ovule. And so these do not follow simple Mendelian rules. And so if we look at a pedigree of something going through mitochondrial, like a gene or trait that's inherited by the mitochondria, we can see that if mom has it, dad does not. We can see what happens. Okay, all of the daughters have this allele too. And not only that, but the daughter's kids all have it as well, right? So that's 100% inheritance rate because the females are passing along the mitochondria, so they're passing on that trait. If, for example, we had a hypothetical male, would it be shaded or not shaded? Should be shaded. But if he married a female that does not have it and they have kids, would you predict them to have it or not have it? They should not have it, right? Because, again, it's the female that's carrying along that mitochondrial trait. So males can't pass it on, and that's what we see down here as well. Only females. And so those were a few different types of non-Mendelian inheritances, and I hope that was helpful. Thanks.